I would like to thank the organizers for giving us an opportunity to present our technology. I kind of cheated a bit because I'm localized in Montreal, so I didn't, but I'm traveling a lot on the West Coast. So biOasis Technologies Inc. is a public company. It's traded on the TSX, and so for me, being a scientist, it's kind of, hold on. I have to show this forward-looking statement. And so, I mean, to go to the specifics, I mean, what we're doing, and it's something a bit different from vaccine development, is we're looking at developing new ways to get therapeutics delivered across the blood-brain barrier to the brain. So everybody knows that that's quite a challenge, especially when you treat people that are affected by neurologic diseases, neurodegenerative diseases, brain cancer, or other things. You need to cross the blood-brain barrier, and as you can see here, that's the and why this blood-brain barrier exists is because it's a protection for nerve cells, for other brain cells, and the protection is basically in place because you have a tight junction between endothelial cells, so a compound cannot cross this, go to the tissue by going in between the cells, which it does in normal capillaries. And also you have lots of resistant factors, MPR, MRP, PGP molecules that are basically efflux pumps that get everything out of the brain. This is just a characterization of it. And then you have a representation that everybody in the field basically shows is how the brain is perfused. It's the highest density of vasculature in the brain, composed in venules on the surface and then capillaries inside. So if you can cross the blood-brain barrier, the advantage is you don't have to migrate long distance to get to the first targets. I mean, the minimal distance is 20 microns. So you can imagine that if you can get a way to get drugs across the blood-brain barrier, it's going to be much easier to treat brain diseases. So that's basically our main concern at the biOasis, and that's what we are doing. So that's the problem. I mean, the medical field, if you have something that crosses the blood-brain barrier, you basically, it's much more, treatment are much more easier, and many neurological conditions such as AD, Parkinson, brain cancer would be easier to treat. I don't have to say that to this audience. I think it's a large unmet market. I mean, the market size is something like $120 billion per year in 2015, and that's with drugs that are not working very well. So as soon as something going to be developed to get the treatment easier, is going to be a much bigger market. So what we have at biOasis, we call it jump to it. We call it Transcend. So there's a protein vector that has been proven that transports drugs across the blood-brain barrier, and it can transport large and small compounds. And what is very important is it can reach therapeutic concentration in the brain. So I'm not going to go to all the data we have generated, but I mean, I can speak about it. I can show you that at one-to-one meetings. We have shown that with small chemotherapeutics, we can reach therapeutic concentration in the brain. And so far that has been done with mice implanted with brain tumors. And then we have done it with lysosomal enzymes. So we can not only go across the blood-brain barrier, but we can go to specific compartments in the cell of the brain. And I'm going to show you some data on antibodies. So I mean, I think it's very adaptable to large molecules, to biologics. So what we have a strong IP position. We generate new IP every time we continuously. We have a good approach with companies to do feasibility study with their own product and to go into the evaluation of what's the concentration, what's the percent injected dose that you can get in the brain. And science leadership team are going to go that at the end. And so we have the potential to bring changes in the medical field. So what we're concentrating on, the internal programs are basically based on what we do best, what we have some concrete evidence that it's going to work, is a lysosomal storage disease. And so we have entered a collaboration, a feasibility collaboration with Shire AGT in Boston for enzyme replacement therapy. That's a big need because lots of lysosomal storage disease are affecting the brain. If you don't get the enzyme in the brain, you maybe treat people with enzyme replacement therapy now, the enzyme not going into the brain. In 20, 25 years, you maybe find out that these people treated develop neurodegenerative diseases. So that's an important point that we're addressing now with Shire. Brain cancer, we started doing looking at Herceptin, at anti-HER2 an antibody that has been shown to be very efficacious in the periphery for breast cancer, but people come back with brain metastasis. So that's something that we want to address, and we are actively working on that. We have some data for that. We do a quantification, and we have some collaboration with a group in Texas Tech University that has an animal model for brain metastasis. And then recently we just showed that an antibody against anti-A beta peptide antibody can cross the blood-brain barrier. So is that a potential treatment or not? That's another question, but people in companies tend to tell me that if we can go into the brain, it's more efficacious. But I mean, we're concentrating our most advanced program at this level internally. So an example here, the way we do it is we label antibodies with fluorescent markers. So here in green you see the Herceptin labeled with rhodamine, but conjugated to what they call to transcend. And so you can see the conjugate labeled with rhodamine within the brain parenchyma. In green, in blue, you have the DAPI blue that labels the nuclei of the brain cells. In red, you see the capillaries labeled with CD31. So you can see here that the majority of the blue is located in the brain parenchyma, and we have done multiple experiments with that. We have done that with other markers and so on, and found that our conjugates normally cross the blood-brain barrier very rapidly. And if you do a quantification 24 hours after injection, you get over 10 times more conjugates with the antibody in the brain parenchyma. Our external programs, so we're concentrating on lysosomal surgeries, oncology. An external program we are discussing with companies to do the feasibility studies, research collaboration, and we're pretty flexible on the way we design the experiment. Is basically what we do. Basically, is look at fluorescinated labeled with fluorescence markers and look at quantification. So that's what we're doing. Opportunity, but I mean, I don't have to go to the specific with that. Our know-how, it's our experience with the protein with Transcend, and also the IP, and we file IP continuously. This is the group, the management group, and the board of directors. Basically, the people mostly involved in science are Chris Fippiger. He used to be the head of neuroscience at Eli Lilly, BioVale, and Amgen. Wilfred Jeffries is a professor at the University of British Columbia. Myself, I used to work in different companies. Among them, I used to be CSO of Angechem in Montreal. I'm called GABA, so that's just an easy way to say Reinhard Gabatuller, it's GABA. And the third person is a professor at the University of Victoria. So that's it. So we're looking ahead. I mean, what we plan to do is multiple collaboration with companies, putting in together feasibility studies, because our approach is maybe not work efficiently for everything. And actually we have already collaboration with NRC. Our work is done by contracting out. It's, we are 4 people actually in the company, and what we do is looking at people doing outside contract to do all the experiments. So this is again a repeat of the previous slide. So this is just a view of Vancouver, which is not a bad place to have a company. Pretty nice when it's sunny, you know. Problem it's rainy sometimes. Yeah. So that's contact Rob at biOasis.ca. That's our CEO. So and hopefully I can some fruitful discussion this afternoon.